So in this video, we are going to break down two different experiments. The first part is going to be looking at a differential blood cell count. And the second part of this video is going to function as blood typing. And so let's take a look at what we're looking at today. So for our differential blood cell count, the purpose is to determine the percentage of various white blood cells, what are called leukocytes, in a blood sample. And so this basically is a way to give physicians an idea of what is going on in the body. Meaning, is there a sign of infection? Is there an allergic response happening? What is going on? And so it's a way to look at the relative proportion of particular types of white blood cells. So when we talk about leukocytes or white blood cells, they can be broken down into two categories. One category is referred to as granulocytes. And granulocytes have prominent cytoplasmic granules. So they have these distinct little granules within their cytoplasm that do a variety of things. So in the category of granulocytes, we have neutrophils, eosinophils, and basal fill. The way that I remember this is if it says fill, it's filled with granules. So fill means that it's filled with granules and it's a granulocyte. We also have agranulocyte. A means without. So that means that these are cells that lack those cytoplasmic granules. So for this category, we have our lymphocytes and our monocytes. And so we're going to talk about what each of these different types of white blood cells do. So what you're looking at is a normal range and percentage for men and non-pregnant women. Now, what you want to realize is this is a rough guideline of what the percentages should look like. But what you really need to realize is that this is variable. Some books are going to list neutrophils as being 60 to 75%. Others might list it as being 55 to 65%. So there is quite a bit of variability that can happen with this. And so just be aware of that. And so what we're looking at is that if we were to separate the blood into distinct fractions, we would have our red blood cells. So here on the bottom of the tube are the red blood cells. We would have white blood cells, and we would have the plasma, which is the liquid portion of the blood. That's what's going to contain the clotting factors, for example. So if we were to look at our white blood cells, the easiest way for me to remember the relative abundance of these different types of white blood cells is to remember never let monkeys eat bananas. So this is how I remember this. So never let monkeys eat bananas. And so you want to know the relative abundance. Now, do I care if you memorize the exact range? No, I don't. I'm not going to ask you a question that tries to trick you by putting an answer choice that is 60 to 70% or 55 to 65%. That's not the point. The point is the relative abundance. So neutrophils are going to be the most abundant, followed by lymphocytes, monocytes, eosinophils, and basophils. So notice that for basophils, they're pretty rare when you're looking at a blood smear. Basophils of the white blood cells, basophils make up only 0.5 to 1%. And so what you're going to see when we look at these blood smears is that in any given field of view, maybe you're going to see, 2 or so white blood cells. If you're looking for a basophil, right, you have to see at least 100 white blood cells before it's likely that you're going to find a basophil because they only make up 1% or 0.5. Maybe you have to look up 200 cells or whatever it might be, right? So Basophils are going to be extremely rare. You're going to have to really search if you're looking for a basophil in a blood smear. They're very rare. So we're going to walk through and talk about the different types of blood cells and what they do. So the first white blood cell that we're going to talk about is going to be the neutrophils. Because again, never let monkeys eat bananas. Neutrophils are going to be the most abundant. Now again, seeing that the name says fill tells you that this is a granulocyte. It's filled with granules. And we'll talk about what those granules do in just a moment. So in terms of relative abundance in the blood, these are going to be the ones that you see the most often. So between 55 to 65% of white blood cells will be a neutrophil. So by far, this is going to be the one that's the most common. In terms of diameter, the diameter of a neutrophil is between 12 and 15 micrometers or microns in size. Now, why am I telling you that? Well, because the size of the white blood cell sometimes can help you with its identification. Red blood cells are approximately 7.5 micrometers in size. If you remember when we talked about bacteria are typically between 1 to 2 micrometers in size. So red blood cell is about 7.5 micrometers in size. So if you compare that red blood cell to the size of the neutrophil, you'll notice that the neutrophil is almost double, or in some cases more than double, the size of the red blood cells. And so the neutrophil is going to be quite a bit larger relative to the red blood cell. Now, in terms of the nucleus is the darker stained part. These blood smears can be stained using what's called right stain. And so the nucleus is going to be the darker stain. Now, in the nucleus, in the neutrophils, it can have between 2 to 5 lobes, meaning that it can be segmented into several parts. Now, when we talk about neutrophils, there are some terms that we use in terms of if they are a mature neutrophil or an immature. If they are a mature neutrophil, we call them a seg because they have a segmented nucleus. An immature neutrophil is going to be referred to as a band because its nucleus is not yet segmented. So if we look at this top picture here, this is going to be a mature neutrophil. Down here on the bottom left, what you're seeing on the left is an immature neutrophil. Its nucleus is not segmented yet. It's a band. So both of those are going to be neutrophils. So neutrophils can vary in the way that they look. And so because they can vary in the way that they look, they are what is called polymorphonuclear neutrophils. Polymorphonuclear neutrophils. So they can have a variation in their shape. So again, they're both neutrophils. Some are segmented, some are bands, but they're both going to be our neutrophils. So in terms of our cytoplasmic granules, the cytoplasmic granules in a neutrophil, again, if it's filled, it means it has granules. A neutrophil has cytoplasmic granules, but they stain poorly, meaning that in the cytoplasm, in this part, it's difficult to see where those granules are. They are not as abundant as an eosinophil or a basophil. For those both stain more distinct. But in the case of the neutrophils, they're not very distinct. The granules for a neutrophil is that they're going to contain antimicrobial chemicals. So they're used to fight off foreign invaders. Now, the function of neutrophils is that they are phagocytic. They're able to do phagocytosis. That means that they're going to, again, send out those extensions and engulf the bacteria, for example, and take it in and destroy it. And so you might recall back in our lecture on cells, I showed you a video of a white blood cell chasing a bacteria. And remember that when the white blood cell got to the bacteria, it did phagocytosis and it engulfed it. So that's phagocytosis. So function of the neutrophils, phagocytosis, and digestion of usually bacteria. Now, it's not always bacteria. It could be other foreign invaders, but usually bacteria. And neutrophils are going to be our first responders. They are the ones in an infection that are first on the scene. And so depending on the abundance of those neutrophils tells us something about the patient's health. So if they have an increase in neutrophils, that's called neutrophilia. Usually indicates an infection. So if we see a patient who has an increase in the number of neutrophils, that again is referred to as neutrophilia, and this is usually an indication that the patient has some sort of infection. And so that is a good way to know if a patient has some sort of active infection. Now, on the flip side, if we have low levels of neutrophils, we have something called neutropenia. And so low levels of neutrophils is not a good thing, because that means that the immune system is not functioning optimally. And so an example of this would be if, for example, a patient is taking chemotherapy drugs. Let's say they're undergoing treatment for cancer. Well, if they're undergoing cancer treatments, not all cancer treatments are very targeted, meaning they might not only affect cancer cells, but they might affect other actively dividing cells. Blood cells are constantly turning over. And so what can happen is that when patients are taking these chemotherapy drugs and they're preventing actively dividing cells from dividing, what can happen is that patients who are undergoing chemotherapy can end up having neutropenia because they are not making enough of those white blood cells. And so as a result, what that tells physicians, and physicians will monitor this, because if the patient has a decrease in the number of white blood cells, it makes them more susceptible to other types of infections, things that a normal, healthy, functioning immune system would be able to handle. But in the case of somebody who has neutropenia, they are not producing enough of these neutrophils, and as a result, their immune system is not functioning optimally. And so this is for the neutrophils. So again, these are the first responders. Next, we have our eosinophils. Eosinophils never let monkeys eat bananas. Notice it's the second to last in terms of abundance, so it's pretty rare. About 1 to 3% of white blood cells will be eosinophils. And in terms of their diameter, they are between 12 to 15 micrometers in size. So basically, for all of our granulocytes are all about the same size. They're about 12 to 15 micrometers in size. You're going to see later that the agranulocytes are the ones that are going to be a little bit different. But for these, they are approximately the same size, between 12 to 15 micrometers in size. In terms of the nucleus has 2 lobes. Notice it kind of looks like little alien eyes. Each of those little dots, those purple circles, are going to be the lobes of the nucleus. Cytoplasmic granules, they're present and they stain red. So notice you see these little red circles within here. That the name eosinophil comes from the fact that the eosin is a red dye, just like the granules are red. So in those cytoplasmic granules contains antimicrobial chemicals as well as histamine, which is a moderator of inflammation for an allergy response. So the function of the eosinophils is that they are phagocytic. So again, they're able to do phagocytosis. They are used to fight helminth infections, worm infections. So they're used against helminth infections, and they play a role in the allergy response. So again, if we see an elevated number of eosinophils, it might indicate that the patient is trying to fight off or is undergoing an allergic response. And so those are going to be our eosinophils. Basophils. Basophils, again, most rare, between 0.5 to 1%. The diameter is 12 to 15 micrometers in size. In terms of the nucleus is either unlobed. Notice that it's in this cell, it's this dark purple here. So that's unlobed, or it could potentially have 2 lobes. Cytoplasmic granules, that's the key for identifying the basophils. The cytoplasmic granules are going to stain dark purple. So you're looking for those dark purple dots. That's what's going to tell you basophil. Those granules contain histamine and other chemicals as well. And so the function of the basophil is that it's used to release histamine during an allergic response. So both eosinophils and basophils play a role in the allergic response. Now we're moving on to our agranulocytes. So these are going to lack our cytoplasmic granules. And so now we're looking at our lymphocytes. So in terms of their abundance, never let, so they're the second most abundant. So about 25 to 33%, so about 1/4 to 1/3 of the white blood cells would be lymphocytes. In terms of diameter, they're quite a bit variable. They can be between 7 and 18 micrometers. Notice 18 says rare. Most are 10 micrometers. So for me, the way that I remember, or the way that I identify a lymphocyte, I think of lymphocyte little. These are the little white blood cells. Notice if you compare this white blood cell versus this red blood cell, notice that the size is not double, right? It's only slightly bigger than the red blood cell. And so that's one of the things that you want to look for when you're looking for a lymphocyte. Lymphocytes are little. In terms of their nucleus is spherical, so it's round. It has very little cytoplasm. So notice that the nucleus occupies most of the space within a lymphocyte. Cytoplasmic granules, again, they are absent because, right, they are not granulocytes. These are our agranulocytes. So these lack the cytoplasmic granules, and the function of the lymphocyte is for specific acquired immunity, meaning this is the branch of your immune system that targets a specific pathogen. It's going to fight off a very specific pathogen. And so within the lymphocytes, we have B cells and T cells. B cells produce antibodies. That's their job. T cells do something a little bit different. They're used to direct an immune response. T helper cells, for example, are the commanders of the army. They're the white blood cells that direct the rest of your specific acquired immunity. They help direct your immune system and tell it what to do. So B cells make antibodies, T cells direct the immune response. Now, in terms of looking at these lymphocytes, just by looking at them, there's no easy way to say those are B cells and those are T cells. So we can't tell what's what. All we can say is that these are going to be our lymphocytes. They also, within this, we have natural killer cells. And these kill foreign or infected cells without antibody, antibody, antigen interaction. So normally your B cells produce antibodies, and those antibodies are highly specific, and they recognize an antigen. An antigen is a molecule on a foreign invader that stimulates an immune response. So this is what gives your immune system some specificity, is because they target different antigens. Natural killer cells can kill foreign or infected cells without that antibody-antigen interaction. They work a little more non-specifically. And so we'll learn more about those in our immunology lecture. So those are our lymphocytes. Next, we have our monocyte. Never let monkeys, so the third most abundant. They are about 3 to 7% in the white blood cells. In terms of their diameter, these are about 12 to 20 micrometers in size. So these are the largest ones. So these are going to be very large in size. In terms of the nucleus has this kind of horseshoe or kidney bean shape. You can still see the cytoplasm, so cytoplasm is still prominent. In terms of cytoplasmic granules, they are absent, right? They don't have those cytoplasmic granules because, again, they're not a granulocyte. Their function is that they are used for phagocytosis. Monocytes will differentiate, meaning they'll become specified to become a cell called a macrophage. And a macrophage is going to be a phagocyte. It's a cell that does phagocytosis for foreign invaders. And so these are going to be your monocytes. So this is just showing you some different pictures of different types of white blood cells. And so again, the top are going to be the granulocytes and the bottom are the agranulocytes. And so you can actually see the size difference when they draw them to scale. So neutrophils are going to have this possibly segmented nucleus, which has between 2 and 5 lobes, right? So here's one lobe, here's another. So this one has 2 lobes. We have our eosinophil. Again, it kind of looks like our little alien eyes. It has 2 nuclei. And the key for this is the red cytoplasmic granules. We have our basophil. Basophil have the dark purple cytoplasmic granules. When we get to our agranulocytes, this bottom picture is going to be a monocyte. It's large, it's very massive, it has that horseshoe or kidney bean shape nucleus, and it lacks cytoplasmic granules. If I look at a lymphocyte, again, lymphocyte little, notice that if you compare the lymphocyte versus the red blood cell, it's not significantly larger, right? Its size is closer to that of the red blood cell. So what you're looking for in this is that the nucleus is going to be very prominent. It's going to take up most of the cell, and the cell is going to be smaller than the other types of white blood cells. And so that's what you're looking for when you're trying to look for a lymphocyte. So I have a video to show you that is going to talk about flow cytometry.
In this animation, we will introduce you to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique.
So what you saw was that the flow cytometer is an automated way to count cells. And so what happens in our flow cytometry is that we have a laser beam that is emitting light. And that light is going to hit the cell, and it's going to cause that light to diffract. So we have our photodetector, and what happens is as the light hits the cell and it refracts, what we call the forward scatter, meaning the detector that's straight across, is dependent on the size of the cell. So the larger the cell, the more the light is going to diffract, and we get a higher forward scatter. So notice on this axis, we have our forward scatter. And so low forward scatter would be our lymphocytes, We have our monocytes and our granulocytes are approximately the same size, so we see them with approximately the same forward scatter. So then if they have a similar forward scatter, how do we differentiate the monocytes from the granulocytes? And that is that we detect something called side scatter. And so there's another photodetector that is perpendicular to where the light is going. So the light's trying to pass through, but we also have a photodetector that is perpendicular to that. And we call that side scatter because the light's going to the side. In terms of side scatter is dependent on the complexity of the cell. For example, if we have a granulocyte and it has lots of cytoplasmic granules, well, when the light hits those granules, it's going to cause the light to bend, and we're going to get a lot of side scatter. And so notice that if we look on this axis and we look at side scatter, the high side scattered would be the granulocytes. Then we have our monocytes, and our lymphocytes would be less. And so what we get is we end up with 3 distinct pools. So on the bottom is just another way that you would actually see this on the computer. Each of those little dots represents one cell. So that's one cell's forward scatter and its side scatter. And then we define parameters saying this amount of side scatter and this amount of forward scatter would be a neutrophil, for example. And this amount of forward scatter and this amount of side scatter would be a monocyte, and so on and so forth. And so this is just an automated way to count the number of white blood cells. Because if you think about it, if you were doing this the old-fashioned way, and you took a blood smear and you looked at the blood smear under the microscope, right? What you see is in that field of view, we view those slides under 1000x total magnification. And when we view those slides, we don't see a bunch of white blood cells in our field of view. And so historically, the way that this would be done would be to actually count the number of white blood cells of each particular type. Now, that's time consuming, it's tedious, it's hard to keep track of where you are, it's not ideal. So using flow cytometry is a way to automate differential blood cell counts and to use this machine to help quantitate the number of different types of white blood cells. And so this is going to be our flow cytometry. So what I want you to do is I want you to practice and identify the following blood cells. So A is this one here, B, C, D. This is E, this little one, F. So pause your video, and once you're ready, you want to turn it back on to listen to the answer. Now again, you want to work through this yourself first because you're going to get the most out of it by attempting to do it yourself. So pause your video. When you're ready, push play. So let's talk about A. What type of white blood cell is A? Answer, A is a neutrophil. It's an immature neutrophil that's called a band. What about B? What type of white blood cell is B? Lymphocyte. Lymphocyte is little. Notice how much smaller, especially next to C. Look at the difference in size. So B is a lymphocyte, little. The nucleus occupies most of the space in the cell. C is going to be which one? Answer, monocyte. It has that kidney bean or horseshoe-shaped nucleus. It's very large. D would be an eosinophil. It has the prominent red cytoplasmic granules. E would be the red blood cell, or what we call an erythrocyte. F is going to be a basophil. because it has the dark purple cytoplasmic granules. Now again, the hardest two I think that students get tripped up about comparing is going to be the basophil and the lymphocyte, because sometimes students look at the basophil and they see the purple and they assume that that's the nucleus. It's not. So dark cytoplasmic granules are the key for basophils, but again, also use size to help compare. Lymphocytes, little. If I were to ask you about this one, what would you say? What type of cell is that one? Answer is, it's a neutrophil, right? So that's another neutrophil. That's a mature neutrophil, which again, we call a seg, for segmented nucleus. So the next part of this is just showing you some different examples of different types of white blood cells. So first we're looking at our neutrophils. So here are the neutrophils. Again, they're quite a bit larger than the red blood cells, but you can see the segmented nucleus. Again, the granules don't stain well, so you're not really able to detect by this that they're a granulocyte, but they are a granulocyte. But this purple that you're seeing there is going to be the nucleus. And again, if we were doing this in lab and you were looking for these, this would be the one you'd see the most often. Look at your field of view. There's only one blood cell in that field of view. They're pretty rare. So most of the time when you're looking around on your slide, most of what you're going to see under the microscope would be a neutrophil, because again, they're the most abundant. About 55 to 65% would be our neutrophils. Next, we have our basophil. So here are two pictures of a basophil. So notice that you can see with the arrow, those are the basophil. Again, those have the dark purple cytoplasmic granules. Compare that to this cell down here. If you were trying to identify that cell down there, what would you classify that bottom cell as? Answer, lymphocyte. Notice it could be a little deceiving. If you look at the basophil up here and you compare with that lymphocyte, they look very similar, because again, that nucleus of a lymphocyte taking up most of the cell almost appears the same as the basophil. But a big difference is the size. Look at that lymphocyte relative to the basophil. The lymphocyte is much littler. Its size is more comparable to the red blood cells. Nevertheless, notice lymphocytes are the second most abundant. So you're more likely to see a lymphocyte than you would a basophil, because again, a basophil is only going to be about 0.5 to 1%. They're pretty rare, but you're looking for the dark purple cytoplasmic granules. If you're looking at a lymphocyte, okay, lymphocyte is little. So here are some examples of some lymphocytes. Again, there was also one on the slide before. Their size is more comparable to that of the red blood cells, and their nucleus occupies most of the cell. Here are two examples of eosinophils. So notice that the two nuclei don't always look the same. So in that one picture, they looked kind of like how you see people draw alien eyes. It doesn't always look that way. Notice this one looks a little bit different, but there are two nuclei, and the key are going to be the dark red cytoplasmic granules. That's what tells you eosinophil. So both of those cells would be an eosinophil. And then lastly, we have our monocyte. Monocytes are going to be large, right? They're very large. They have that kidney bean or horseshoe shaped nucleus. What would you say this one over here is? Again, it's a lymphocyte, right? Lymphocyte little. So you want to practice looking at these white blood cells and trying to identify them if you were shown an image of the white blood cell. And so this is going to be our white blood cell, our differential blood cell counts. So for our blood typing, the purpose is to determine the blood type of a person. And so what that means is that we're trying to determine what blood type somebody is. And so when we look at blood types, we have 4 what are called phenotypes, or 4 blood types. We have blood type O, A, B, or AB. Those are our 4 phenotypes. Now, phenotype or physical characteristic is determined by genotype, right? The genes that somebody has is going to dictate what their phenotype is. So let me explain and kind of walk you through how do we determine somebody's blood type. So we need to understand a little bit about how blood types work. So if somebody has blood type A, they have an A-type carbohydrate on the surface of their red blood cell. If they have blood type B, that means that they produce a B carbohydrate. So A produces this A-type carbohydrate, B produces this B-type carbohydrate. If somebody is AB, notice that they have the A carbohydrate and they have the B carbohydrate. They make both. For blood typing, the A and the B, we call those alleles. Those are alternate versions of a gene. They both are going to produce an antigen on the surface of the red blood cell. They're both going to produce this carbohydrate, but the A carbohydrate is a little bit different than B. Now, if we look at A and B, we call A and B, they are what we call codominant. So in genetics, we call this codominant, meaning if you have the allele, if you have the gene for A and you have the gene for B, you're going to express both. You're going to see both. So the A carbohydrate is produced, the B carbohydrate is produced. Now, if we have somebody that is blood type O, O means that the person does not produce A carbohydrates and the person does not produce B carbohydrates. So you can think of their red blood cells as being naked. They don't make A, they don't make B. So if we talk about genetics, capital letters are dominant. Recessive alleles are going to be lowercase. So notice that for type O blood, the genotype is little I, little I. Little I, little I. So in order to be type O blood, you have to have the 2 recessive alleles. You have to be little I, little I. That is going to make red blood cells that are naked. They don't have an A carbohydrate on them. They don't have a B carbohydrate on them. So what that means is that if I am type O blood, I don't make A carbohydrates on my blood. My blood doesn't make A carbohydrates. So my body is going to make anti-A antibodies. Because if I'm O, my body sees A as being foreign because I don't make an A carbohydrate. If I'm blood type O, that means that I produce anti-B antibodies because B is seen as foreign as well. If I'm O, I don't make B carbohydrates. So somebody who is O makes antibodies that are anti-A and anti-B. So what that means is if we think about if somebody's blood type O and we're thinking about what type of blood they could receive if they were undergoing a blood transfusion. So this is showing you this top part is those are the donors. And so if a donor is type O and they donate blood to somebody who is O, blood is fine. So somebody who is O can receive type O blood. However, if the recipient is O and the donor is A, if somebody who's type A blood donates and gives to somebody who's O, we get what's called a heme agglutination. And you'll see this on the next slide, meaning that the red blood cells will clump together. The reason that somebody who's O cannot receive A is that, notice again, this person who's O makes anti-A antibodies. So in their blood, this O recipient, they're producing those anti-A antibodies, and that is going to interact with the A donor, because this A donor, so this would be like the A donor over here, this A donor has the A carbohydrate. If it has the A carbohydrate, that's going to interact with the anti-A antibodies. If the donor is B, right? If somebody who's type B gives blood to somebody who's O, that means that this B is also going to agglutinate, because that B is seen as foreign. Because again, if you're blood type O, you make anti-B antibodies, and that would interact with B. If the donor is AB and they give blood to somebody who's O, again, we're going to get agglutination because both A and B are seen as foreign. So notice, if you are type O blood, the only blood that you can receive safely would be type O blood, because O would be seen as self, and A is foreign, B is foreign, AB is foreign. So if you're blood type O, your genotype is i. You have to have the recessive alleles, and the only blood you can receive is going to be O, because it's the only one that's seen as self. So if somebody is blood type A, again, if they're blood type A, it means that they make the A carbohydrate. Now, in terms of their genotype, they could be what we call homozygous. Homozygous means the same allele. So they could be homozygous for the A, which is, we write their genotype IAIA. Now, you might wonder, why do we write it a capital I and then this superscript, this letter on the top? The reason for that is, notice for AB, remember that I said that A and B are co-dominant? We can't write one as an uppercase and one as a lowercase, because both of those genes are dominant to one another. So the A allele is dominant, the B allele is dominant, the only one that is recessive is the O. So the O is given the lowercase, the A and the B alleles are written as uppercase. So that's because they're co-dominant. So somebody who is blood type A could be IAIA. They could be two A alleles. Or they could be IA little I. So that means that they inherit one A allele and one O allele. Remember that O is recessive to A. O allele basically means they don't make that carbohydrate on their blood. So you're still going to see type A blood because they still have A gene for A. So they are what we call heterozygous. They have IA little I. So either of those two genotypes, IAIA or IA little I, either of those two are going to give you A blood type, meaning that they make A carbohydrates on their blood cells. Now, if you are type A, right, if you're type A, are you going to make in your blood, if you're type A, are you going to make antibodies that are anti-A? And the answer is no, because A is seen as self. A is self. So in that case, right, if you're blood type A, your body is not going to produce anti-A antibodies because A is self. But you will make anti-B antibodies because B is seen as foreign. A is self, B is foreign. So if you are type A and you're getting a blood transfusion You can receive O because, again, O is naked. There's nothing on the surface of the blood cell for the body to react as it being foreign. So you can receive O. You can receive A because A is seen as self, but you can't receive B and you can't receive AB because in both cases that B carbohydrate would be seen as foreign because if you're A, you make anti-B antibodies. If you are blood type B, you have two possibilities for your genotype. You could be I, B, big B, I, big B, or you could be heterozygous and have a B and an O. Both of those are going to give a B type carbohydrate. So IB or IB little I, both are going to give B type blood. And that means that for you, I'm one of these people, I'm type B blood. I produce a B carbohydrate on the surface of my red blood cells. So I produce the B carbohydrate. So in my body, am I going to make anti-B antibodies? Answer is no, because B is seen as self. So I myself don't make I don't make anti-B antibodies. Do I make anti-A? Yes, because A carbohydrate is seen as foreign. I don't normally make A carbohydrates, right? So my body would produce anti-A antibodies. So for me, who's type B, I can receive O blood because O blood is seen as self. I can receive B blood because B is seen as self, but I can't receive anything with an A. So I can't receive A, I can't receive AB. Both A and AB would be seen as foreign, right? So both of those would be seen as foreign because my body would produce anti-A antibodies. So I can't receive anything with an A. If somebody is blood type AB, remember, A allele and B allele are codominant. They're both expressed. So the genotype for this has to be IA, IB. You have one A allele and you have one B allele. Is it possible to have more than two? Answer is no, because all of your chromosomes are paired. Half of your chromosomes came from your mom, half of your chromosomes came from dad. So if you are AB, one parent gave you the A, one parent gave you the B, right? So A and B are codominant. So if you have an A allele and a B allele, you express both carbohydrates. Both of those are seen on the surface of your red blood cells. So what that means is if you're AB, do you make anti-A antibodies? Answer is no. A is seen as self. Do you make anti-B antibodies? No. B is seen as self. So notice for antibodies in the blood, none. There's none because A is self and B is self. So if you are AB, you can receive blood from somebody who is O, because O is naked, there's nothing to see as foreign. You can receive blood from A because A is seen as self, the A carbohydrate, there's nothing foreign. You could receive blood from B because, again, B carbohydrate is self, or you could receive AB. So notice that if you're blood type AB, you can receive blood from anybody. You can get O, A, B, or AB. So we have what we call the universal donor and the universal recipient. So of these four blood types, which one can give to everybody? And the answer is O. Of these four blood types, which one can receive any blood type? Answer is AB. Now I'll come back to the Rh factor in a minute. We'll talk about positive and negative in just a minute. But O can donate to anybody. AB can receive blood from anybody. And so that's why when you watch, medical shows, ER, Grey's Anatomy, and they're in an emergency trauma situation and they need blood, you'll hear them go, I need O-neg stat. They need O-negative blood because if there's not time to blood type the patient, well, as a default, they're going to give O-negative blood because O-negative blood should be able to be received by anybody. O blood is the universal donor. It can give to anybody. Anybody can receive O. But if you're AB, you can only donate to AB. It's the only person that you can donate to, but you could receive any blood. You could receive O blood, A blood, B blood, or AB. So now, critical thinking question. I was trying to track how blood types were inherited in my family. I know that I'm B. I know that my mom is AB. My grandma, which is my mom's mom, my grandma tried to tell me that my grandma was O. So I want you to think about for a minute, is it possible for my grandma to be O and to have my mom who is AB? So think about that for a minute. Is that even possible? Answer is no, it's not possible. Because if my grandma is O, she is little I. My mom, who is AB, is IAIB, which means one of her parents gave her the A allele, one of her parents gave her the B allele. It's impossible for my grandma to be O because my grandma had to give her either the A or the B. Come to find out, my grandma was just wrong. She's blood type B, which makes a lot more sense. But it's impossible. So somebody who is blood type O cannot have an AB child. Can a parent who is AB have an O child? Answer is no. That's not possible either because if somebody is blood type O, they had to get a little I from both parents, right? One allele comes from mom, one comes from dad. So let's ask another one. If mom is A and dad is B, is it possible to have an O child? Mom is A, dad is B. Answer is yes. It is still possible to have an O child because mom could be IA little I, dad could be IB little I, and by chance they could both give the little I and have a kid who is blood type O. So that is possible. You can have two parents that are A. They can have a type O child. What about, can you have a situation where you have two A parents, so two parents who are blood type A, and they have a child who is blood type B? Is that a possibility? Answer is no. Because if the child has a B allele, they had to get the B from one of the two parents, which means that there's no way both parents could be A. A parent could be a B, but you can't have two parents that are A and then have a B child. That doesn't work. But it can happen with the O because O is recessive, right? And so you can have two parents that are not O and then have an O child. But that doesn't work if you have an A parent and a child that's B. That does not work. And so this is just to give you a little bit of understanding about what it means to have these different blood types and what blood types you can accept if you are receiving a transfusion. So what I want you to think about when you think about this is that what you're seeing here is not how we're determining blood type. So don't let that figure with the agglutination here Don't let that throw you off for what I'm going to go over in the next part. They're two separate things. This is not for determining blood type. This is if these are the recipients, over here, which donor blood could they receive? And that is, if you're O, the only donor blood you can receive is O. Because A would be foreign, B would be foreign, and AB would be foreign. So that's what this table is telling you. It's telling you if you're this blood type, if you're this recipient, which blood could you receive from a donor in a transfusion? And again, that's going to come back to the next part, which is going to talk about, well, how do we figure out which blood type you are? How do we know if your blood type is A or B or so on? And so that's the next thing that we're going to focus on. So now let's talk about how do we blood type somebody? How do we determine what their blood type is? And so we need to start by, again, discussing what heme agglutination is. And heme agglutination is a general term applied to any agglutination, so clumping, in which the clumping of the red blood cells indicates a positive reaction. And so if we look at the structure of an antibody has a Y-shaped structure, and it has two binding sites for an antigen. And so an antigen is any substance that elicits an immune response. So what you're looking at is that this antibody here, this is an anti-B antibody. And anti-B antibodies recognize B carbohydrates. So recognize B carbohydrates. Meaning that this anti-B antibody only binds to the B carbohydrate. Now, who would be the provider of this anti-B antibody or anti-B antiserum? Which blood type would have anti-B antibodies in it? And that is, it would be found in somebody who is type A. So recognize B carbohydrate from someone with type A blood. So we get this anti-B antiserum or these anti-B antibodies from somebody who is type A blood. Because remember that if somebody is type A blood, they don't make anti-A antibodies because A would be seen as self, but they would make anti-B antibodies. So now we have our anti-B serum, and next we want to talk about this blood. So this is the blood trying to type, meaning this is the blood that we're trying to figure out what blood type they are. This is our unknown blood. So we want to know what is this person's blood type. So what we do is we take this person's blood, the one that we're typing, and we take that blood and we mix it with anti-B antibodies. Now, anti-B antibodies, right, the antibodies that recognize the B carbohydrate, they don't have the same shape as the A carbohydrate. So notice that the antigen binding site, the site on the antibody where the antigen would bind, has this kind of triangular shape. The A carbohydrate is rounded. And so notice that you don't get binding. The antibody is not going to bind to the blood. And as a result, you're not going to see agglutination. So no clumping. So what that tells us is that if we mix it with anti-B antibodies and we get no agglutination, that means that no B antigens are present. Sorry about the pen, it's behaving funny. So no B antigens are present. So if we do this agglutination Again, we're starting with the person we're trying to type, and we're mixing it with anti-B antibodies. If we get no binding, meaning we get no agglutination, that tells us that there are no B antigens present. So on that unknown blood, there's no B carbohydrates. Now we repeat this, and now we're going to use anti-A antibody. These recognize A carbohydrates. So they recognize the A carbohydrates. So now notice that if you look at the binding site for this antibody, the binding site is round and it recognizes this A carbohydrate on the person we're trying to type. So it's the same blood, but this time now we're mixing it with anti-A antibodies. Again, the anti-A antibodies come from somebody who is type B blood. So type B blood, like I'm type B, in my blood, I would have anti-A antibodies. So my blood could be used to provide anti-A antiserum. So we take this blood we're trying to type, we mix it with our anti-A antibodies, and now because the antibody recognizes the antigen, notice we have binding, and there's 2 binding sites. So what that does is when the antibody binds to those 2 red blood cells, because they have the corresponding antigen, notice that it brings those red blood cells together. It's what we call agglutination. We're getting clumping because that antigen is present. And so if we look and we see this agglutination, that means A antigens are present. So our A antigens are present. So this person has A antigens, but not B. So what that tells us is that the person is type A blood. They have the A carbohydrate, but they don't have the B carbohydrate. And so this is how we would determine somebody's blood type. We take their blood, we mix it with anti-B antibodies, we mix it with anti-A antibodies, and we determine which carbohydrates are on the blood. If that carbohydrate is on the blood, well, that tells you that that is part of their blood type. So let's walk through and fill out a table for each blood type where we expect to get agglutinations. So in this experiment, this top row, it's blood to be determined. Meaning that this is the blood that we're trying to type. We want to know what their blood type is. Now, if we mark plus, that means we expect an agglutination. We expect to see clumping. If we mark negative, that means no agglutination. All right, so let's walk through this. So if the unknown blood is type A, so if the blood that we're typing is type A, if we take that type A blood and we mix it with anti-A serum, which is basically anti-A antibodies, will the anti-A antibodies react with the A blood? And the answer is yes, because remember that this blood, is going to have these A-type carbohydrates. And so if we mix it with antibodies, we'll do this, they're rounded, right? If we mix this anti-A antibody with the A blood, we are going to get agglutination because that antigen is on the blood. Now, if we mix it with anti-B serum, anti-B serum, the antibodies look a little bit different. So they're not rounded, but instead triangular. So will those anti-B antibodies react with the A carbohydrate? The answer is no. So we get no agglutination. For type B blood, what that means for type B blood is that they have B carbohydrates. We have B carbohydrates. So now they have a different shape. So if we have B carbohydrates on the blood cell and we mix it with anti-A antibodies, will the anti-A antibodies bind to the B blood? Answer is no, because this antibody does not have the corresponding shape. So we get no agglutination. We take this type B blood and we now mix it with anti-B serum. Will we get agglutination? Answer is yes, because the anti-B antibodies are going to recognize that B carbohydrate. And so you're going to get binding, and therefore you're going to get agglutination. AB. Remember that AB blood, this pen is being funky, sorry. Here's our AB blood. It has both. It has both A and B carbohydrates on the surface. So if it has both, right, if it has both, and we mix it with anti-A serum, anti-A antibodies, if we mix it with anti-A antibodies, we are going to get agglutination because the A carbohydrate is found on that blood. If we mix type AB blood with anti-B antibodies, we will get agglutination because that means that antigen is found on the blood cell. So we would get agglutination in both for AB. If we have type O blood, remember, is naked. There are no surface carbohydrates. So we don't have our A carbohydrates, we don't have B. So if we mix type O blood with anti-A serum, are we going to get agglutination? And the answer is no. That carbohydrate is not on the blood, therefore no agglutination. We take the type O blood, we mix it with the anti-B serum. Will we get agglutination? Answer is no, because the B carbohydrates are not on the blood. And so this is how you determine the blood types. You're going to look at, so here we have these depression plates and we have these little wells. And so notice that here is well A, down here is well B. We'll talk about RH in a little bit. Don't worry about that one yet. But what we do is we take the person's blood. So this is Mr. Jones's blood. And we put a drop of blood in each of those wells. The one that's A has the anti-A serum. The one that is B is anti-B serum. So notice that in A, we don't get any agglutination. Notice that the blood is clear. You don't see precipitates forming in the blood. So no agglutination. We got no agglutination for A. We mix Mr. Jones' blood with anti-B serum. And notice that you can see this cloudiness here. That's agglutination. So we get agglutination in B. Again, I'll come back to the RH later. For now, just leave that. But if we don't get agglutination with the anti-A, and we do get agglutination for the anti-B, what blood type is Mr. Jones? Well, Mr. Jones has to be type B. And so again, whenever we get agglutination, tells us that the antigen is present. So for type B blood, we're going to expect that we get agglutination for the anti-B serum, but not for the anti-A. If this was A blood, it would be the opposite. We would get agglutination for A and not for B. If the blood is O, we're not going to get agglutination in either. If the blood type is AB, we are going to get agglutination in both, right? So again, look for agglutination. If you see clumping, that tells you that that antigen is present. So if we get clumping in only B, that means that the patient has type B blood. If we get only clumping in A, the patient has type A blood. If we get clumping in A and B, that means they have A carbohydrates and B carbohydrates, which means they're blood type AB. If we get agglutination in neither A nor B, that means that they don't have A carbohydrates, they don't have B, and that person would be determined to be type O. And so this is how we determine blood typing. We do this setup with these antiserums to detect the presence of those antigens. If those antigens are present, that tells you that those carbohydrates are on the surface of the blood cells. And so that is how we would determine blood type. So now let's move on and talk about the Rh factor. And the Rh factor is also referred to as the D antigen. And the D antigen is a type of antigen that can also be found on the surface of red blood cells. So in addition to having the A carbohydrate and the B carbohydrate, it's also possible for red blood cells to have this D antigen, this other surface protein or surface marker on the red blood cells. So this is a clumping factor. And it's referred to as the Rh factor because it was discovered in rhesus monkeys. And so let's walk through and talk about what does it mean if we say that somebody is Rh positive versus Rh negative. So if we say that somebody is Rh positive, that means that they have the clumping factor. They have the D antigen on the surface of their red blood cells. If they are Rh positive, what that means is that they do not produce anti-Rh antibodies. They don't make antibodies against the D antigen because that D antigen would be seen as self. So somebody who is Rh positive will not have anti-Rh antibodies in their blood. If they are Rh positive, they can receive Rh positive or Rh negative blood, because Rh positive would be seen as self. And if the person is Rh negative means that the D antigen is missing. And therefore, it's like, think of Rh negative, think of it like O blood, right? O blood is the absence of the A antigen and the B antigen. Rh negative is the absence of the D antigen. So Rh positive, somebody who is Rh positive can receive either Rh positive because that is self, or Rh negative because there's nothing on the Rh negative blood for that person's immune system to react with. So if you are Rh positive, you can receive Rh positive or Rh negative blood. In terms of your genotype, meaning the genes that you get from your parents, genotype can be heterozygous, so meaning that you have a plus and a minus. So one allele is Rh positive, one allele is Rh negative, but your phenotype would be that you are Rh positive because you are producing the D antigen. Or you could be homozygous for the Rh positive allele. So you could be positive, and that would also make you to be Rh positive because you would produce that D antigen. Let's talk about Rh negative. Rh negative lacks the clumping factor. So it lacks that D antigen. If somebody is Rh negative, they will produce anti-Rh antibodies because, again, for them, the D antigen is seen as foreign. If you're Rh negative, you don't produce the D antigen and therefore could produce the anti-Rh antibodies. If somebody is Rh negative, they can receive Rh negative blood only. It's the only one they can get. They cannot receive Rh positive because for them, if they were to receive Rh positive, they are making those anti-Rh antibodies. And if they were to receive Rh positive blood, they would have an agglutination and the blood would start to clump. So somebody who is Rh negative can only receive Rh negative blood. They cannot receive Rh positive blood. And if somebody is Rh negative, their genotype has to be negative. They have to have inherited 2 negative alleles because the only way that you can be Rh negative is if you are the homozygous recessive, which is the Rh negative. So your genotype would have to be negative. So let me ask it this way. If you have 2 parents who are Rh negative, can they have an Rh positive baby? And the answer is no, because if both parents are Rh negative, right, that means that they have both negative alleles and their kids can only receive the negative alleles. Can you have 2 parents who are Rh positive and have an Rh negative baby? Answer, yes, they can, because those 2 parents that are Rh positive could be positive negative. They could both be heterozygous. And just by chance, they could both give the negative allele. So let's talk about why is it important for the Rh factor. So in addition to blood transfusions, a big problem with the Rh factor can be what's called Rh incompatibility. And so what happens in Rh incompatibility, this happens when mother is Rh negative. So mom is Rh negative. So again, remember that if you are Rh negative, you can produce anti-Rh antibodies because the D antigen is seen as foreign. So let's say we have an Rh negative, right? An Rh negative mom, and her partner is Rh positive. And so one of the things when you're pregnant is that they do determine your blood type. This is how I figured out what blood type I am, because they blood type you when you're pregnant, because they're testing to see if mom is Rh negative. If mom is Rh negative, then they need to know the blood type of the baby's father. Because if baby's father is Rh positive, right? So if mom is Rh negative, she is negative for her genotype. For dad could be plus, or dad could be plus minus. If dad happens to be plus minus and mom, remember, is minus, so let's say dad is plus minus and mom is minus, there is a chance that the baby could also be Rh negative and there would be no problem. However, dad is Rh positive, which means that he has the potential to give that positive allele to the baby. So the problem is if baby is Rh positive. So the problem comes into play if baby is also Rh positive. And that's because what happens is that when the baby is in utero, when it's in the womb, the baby's blood is being filtered by the moms. So first pregnancy, mom's body has never seen the Rh factor, right? And so because she's never seen Rh factor, she doesn't make anti-Rh antibodies. However, when delivery occurs, so when mom goes to deliver the baby, what ends up happening is that when we go to deliver, the placenta is going to separate from the female, and mom's blood and baby's blood commingles. And so now mom's body sees that Rh positive blood, and now mom starts to produce anti-Rh antibodies. So now she has her anti-Rh antibodies. Now, first baby is out, no problem. There's no complication for the first baby. It's subsequent pregnancies. So if mom has already been exposed to the Rh positive blood, she is going to make anti-Rh antibodies. Now, those anti-Rh antibodies, when mom makes it, when mom's body responds to an immune response and has been challenged with an antigen, what's going to happen is that that's going to initiate memory cells that remember that Rh antigen. And if she were to come in contact with that antigen again, well, then if she were to come into contact with that, now the antibodies are going to be produced very quickly, and they're going to start to destroy the baby's red blood cells. So again, first pregnancy, baby's out, baby's fine. But again, it's going to be the second pregnancy. So now when she gets pregnant again, and she gets pregnant with another Rh positive baby, her antibodies are getting into the baby and they're attacking the baby's red blood cells. Now, it's not as damaging while the baby is in utero, because during this time, mom's body is again filtering the baby's blood. And so she's breaking down some of those harmful products that could be produced when the baby's red blood cells are being digested. However, when mom goes to deliver baby number 2 and the baby comes out and mom is no longer filtering baby's blood, now the baby's red blood cells are being destroyed and there's no way for the baby to get rid of those products. And so what you end up getting is it causes something called hemolytic disease of the newborn. The newborn's red blood cells are starting to lyse, they're starting to get broken down, and that is going to create these toxic products in the baby. And if not prevented, and the baby's blood is contaminated with maternal antibodies, sometimes what ends up happening is that they have to replace the baby's blood by transfusion of uncontaminated blood, because otherwise mom's antibodies will just keep attacking those blood cells and will damage those red blood cells, and the baby becomes anemic. And so what ends up happening is, and the way that we prevent this from happening, is that mom during delivery, so let's say this is mom with her first Rh positive baby, she's going to get something called Rhogam. And Rhogam is an injection of anti-Rh antibodies. AB is antibodies. So during delivery with the first baby, right? First baby, if the mom during labor gets her injection of this Rhogam, which is these anti-Rh antibodies, those antibodies, when you inject them in, the antibodies will neutralize the baby's blood. And if mom's immune system doesn't have to create those antibodies, if the antibodies are already there to neutralize the baby's blood, then if the antibodies are already there, mom's immune system doesn't make antibodies. And if mom's immune system doesn't make antibodies, then she will not have those memory cells. And those memory cells won't produce those antibodies. And so again, this is called Rh incompatibility. and it happens when a woman is Rh negative and she has a fetus that is Rh positive. If the fetus is Rh positive and that baby's blood mingles with mom's, well, that Rh positive blood is going to stimulate mom's immune system to make those anti-Rh antibodies. And now those anti-Rh antibodies can affect subsequent pregnancies. So again, it's not really the first pregnancy that we're concerned about. It's any subsequent pregnancy after that. If mom has already made those antibodies, it's problematic for any other child. And so that can be prevented though, or at least helped, if mom receives that Rhogam injection. If she gets those anti-Rh antibodies and her immune system doesn't mount the response, well, then she's not going to have those memory cells and it's not going to affect subsequent pregnancies. And so this is how Rhogam works. So going back to our universal donor and our universal recipient, our universal donor is O, is it negative or positive? Which one can give to everybody? And the answer is O, negative. Because if they are Rh negative, negative can receive it, positive can receive it. Because again, negative means they don't have the D antigen and therefore they're not going to have anything to react against. So O negative is the universal donor. In terms of the universal recipient is AB, positive or negative. Which one can receive anything? Answer, positive. AB positive can receive any blood because they can receive AB minus, they can receive AB plus. So if you are AB positive, you can receive any blood type. A positive, B negative, A negative, O positive, O negative. If you are AB positive, you can get any type of blood. O negative, if you're O negative, blood banks love you because they want to have that O negative blood on hand so that in case of a trauma or an emergency, they have that blood if they don't have time to type the patient. They can give O negative blood and not really have to worry about it as much. So if you're thinking about it, if we have a donor and the donor is let's say the donor is A positive. Okay, donor is A positive. The recipient is O negative. Can the recipient get that A positive blood? Answer is no. What if the donor is, let's say the donor now is O positive? Can the O positive blood be given to A negative? O positive to A negative. Answer is no. Because that O positive, yes, the O can be accepted, but the fact that the blood is positive cannot be received by an A negative person. So you not only have to look at the blood types to see if they're compatible, but you also need to make sure that the Rh factor is also compatible. And so that's something to keep in mind when you think about blood typing. Coming back to our determining somebody's blood type. This third well on the depression plate says Rh. That is going to be taking that person's blood, so Mr. Jones' blood, and mixing it with anti-Rh antibodies. So notice that when you mix the blood with the anti-Rh antibodies, you get no agglutination. There's no clumping there. So if we don't have agglutination, right? If we don't have agglutination, is the Rh factor present? And the answer is no. Because if the Rh factor is present, if that D antigen is on the blood, then you would get agglutination with the anti-Rh antibodies. But you don't. So Mr. Jones would be B negative. B negative, because again, that Rh factor is not present on the blood cells. And so that baby or that person, Mr. Jones, is going to be B negative. So let's go through and look at some other examples and see if you can determine blood types. So this is for that last question in your question set. It has you determine the blood type of the following people. So what you're looking at is on the left, we have Mr. Jones. And we already determined Mr. Jones is B negative because we get agglutination in B, but we don't get agglutination in A. We don't get agglutination in the Rh. So that means that Mr. Jones is B negative. Look at Mr. Smith. What I want you to do is I want you to pause and try and do this yourself. Figure out what blood type is Mr. Smith. After you do that, go ahead and push play to hear the answer so that you make sure you're studying the right thing. So if we look at Mr. Smith, did we see agglutination for A? So let me change my ink here so maybe it shows a little better. Did we get agglutination for A? Yes. We see agglutination. We see clumping. We see precipitates in A. Do we see agglutination in B? Answer is no. So no agglutination in B. Do we see agglutination for Rh? Yes, we do. We see clumping there. So what is Mr. Smith's blood type? Well, Mr. Smith has the A antigen. Mr. Smith has the D antigen. So Mr. Smith is A positive. Mr. Smith is A positive. Let's go to the next one. So we have two more people. We have Mr. Green and Ms. Brown. Mr. Green and Ms. Brown. So again, pause the video, look at the following images, and then determine the blood type. And then when you're ready, push play to hear the answer. Okay, so hopefully you thought this through. Mr. Green, we'll start with Mr. Green. Do we see agglutination in A? Yes, we see clumping. Do we see agglutination in B? Yes. Do we see agglutination in the Rh? Yes. So what that tells us is that Mr. Green has A carbohydrates, Mr. Green has B carbohydrates, and Mr. Green has the D antigen. So what would Mr. Green's blood type be? Well, that means Mr. Green is AB positive. AB positive because he has the A carbohydrates, he has the B carbohydrates, and he has the Rh factor. Next, let's look at Ms. Brown. Ms. Brown has no agglutination for A. Ms. Brown has no agglutination for B. And Ms. Brown has no agglutination for Rh. So we don't have the A carbohydrate. We don't have the B carbohydrate. We don't have the D antigen. So what is Ms. Brown's blood type? She is O negative. She is O negative because she doesn't have A or B, and she doesn't have the D antigen, which means that she is O negative. And so this is how we would determine blood type.